Hey everyone, we have spent this series looking at biological data in high resolution, one sequence or one 3D structure at a time. But a cell isn't a bag of isolated parts. It's a complex machine where proteins are constantly talking to each other. So today we are moving into systems biology. We're going to map out protein-protein interaction networks or PPIs for short, and we will learn how to use a powerful library called NetworkX to find the VIP proteins that hold the whole cell together. So let's start this networking. So what exactly is a PPI network? So a protein-protein interaction network is essentially a map. So in mathematical terms, we call this a graph. So the nodes or the points are the proteins and the edges represent a physical interaction between them. So by studying these graphs, we can identify hubs, which are the proteins that interact with hundreds of others. And if a hub protein is mutated, it can crash the whole cellular system, which is why hubs are often major drug targets in cancer research. So we will use a library, which I already mentioned. It's called NetworkX. If you don't have it, you can install it like this. And also you will have to install Matplotlib for visualization purposes. So after installing those libraries, we can get started. So let's first of all, let's start with importing the libraries. NetworkX as NX and import matplotlib dot iplot as plt. So let's build a small network manually to see how it works. So imagine we have five proteins. Five proteins, P1 through P5. So we will start with something like this. We will create a empty graph object nx dot graph. Then we will add interaction edges and interactions is equal to, and we can create something like this. Right. Let's say P1 is a hub interacting with everyone. So and after that, we will do g dot add edges from and interactions. Then we will print the output. So nodes in the network, uh, nodes in the network, g dot nodes. Yeah, this should do it, I think. Okay. Why is it showing an error? Yeah, we have to close this, right? And similarly, we can copy this, and here we can write a number of interactions, and here we can use g dot number of edges, right? So let's try and run this, and there you have it. So as you can see, five nodes are there in the network, and total number of interactions are also five. So how do we mathematically find the most important protein? Uh, so what we do is we look at its degree, simply the number of connections it has, right? So we will run a loop for that for protein and degree in g dot degree. We will print protein protein name has degree connections, right? Okay, so let's try to run this, and there you can see each protein and how many connection it has. So in a real biological data set, you might have 20,000 proteins, and the one with the highest degree are your master regulators. So NetworkX can calculate this for the whole genome in seconds. So Network is in very useful if you cannot see it. So we will use matplotlib to draw our graphs. So first of all, we will do a layout. POS is equal to nx dot spring layout. Okay, then we will draw the network. PLT dot figure figure size. Let's go with 8:6. That should be enough, I think, or slightly bigger, but better for visualization. Then we will draw the plot. Um, G position, and we will use labels. So we will use with labels as true. After that, for labels, we will have add some other properties. Node color, let's go with sky blue. Then node size, uh, let's go with suppose 2000. Then edge color, we can go with gray. And finally, uh, let's add font weight as well. We will go with bold. So these properties should be enough for the plot. Then we will assign a title. Let's say simple PPI network, right? And then we will show the plot, right? So let's try and run this, and there you have it. See how P1 is at the center. That is our hub. In a real study, you would download data from databases like String or BioGrid and load thousands of interactions into a loop and find the essential genes for a specific disease. Mapping the intercom takes us from looking at parts to looking at the whole system. So today we use NetworkX to build, analyze, and visualize a basic biological network. This concludes our first big part of the Bioinformatics for Beginners module. And we have covered everything from basic DNA strings to complex 3D structures and cellular networks. So you might now have a solid foundation in using Python for your preliminary biological research. So, but the journey is not finished yet. We will dive into next generation sequencing and other things in the coming next videos. So, thank you so much for speaking with me throughout this course. And all the code is as usual will be available on GitHub. And if this series helped you, please leave a comment, subscribe, and share it with a few bioinformaticians. So, I'll see you in the next module. Thank you.